right. Okay. That's weird. All right, can I have your attention, please? We're going to go ahead and get started, and for some reason, this is not as sensitive today. Let's try that. I hope it doesn't. Okay. That's perfect. All right, so last lecture. Woohoo! You guys are almost done with cell bio, at least for class. Of course, you're going to need it for the rest of your lives, so haha. I can't get away from it. So what I'm going to do is do a really short half a lecture to finish up the stem cells that we've been running behind on everything. I want to finish by 11:35 sharp so that you have 40 full minutes of review for your final exam and exam 4. And the way we're going to do that is your TAs are going to go through all the 4 units in 10-minute segments. You guys can ask questions. Dr. Lin and I are both here to help answer those questions. So that's what we're going to do starting at 11:35. Because we are running generally behind the semester, I might skip a few slides here and there. And I'm going to be even more explicit than usual about what you need to know for exam 4, because I don't think it's particularly fair 2 days before exam 4 to throw a bunch of stuff at you, right? So we're going to be really simple today in what we cover. It's more of a fun, I hope, half an hour or so to finish off stem cells, and then we'll move on to your review. All right. So this is where we left off, we left off here, where I was talking about stem cell differentiation in a dish. One of the therapeutic mechanisms that you hear so much about in the news right now. Can we take stem cells and turn them into different things? So this slide is to illustrate both the potential and how far we have to go of doing something like this. And guys, there's a lot of rustling and whispering. Please, let's kind of focus here now because we don't have a lot of time. Thank you. Okay, so what I want to emphasize here is that, wow, wouldn't it be cool if you put these cells in a dish and you can change different things in the media and you can make neurons that secrete these different things, dopamine and serotonin, these signaling molecules, or cells that make insulin. Could that be a cure for these diseases? In theory, yes, we can do this. This is from a dish. People are able to take stem cells in a dish and turn them into these different cells. Unfortunately, that's just, let's say, half the battle. Once you have those cells, how do you actually get them to the patient? How are they not rejected by the patient's immune system? How do they go to the right place and do the right thing? How do they not do too much stuff? So there's a lot more to figure out, but we're making progress with at least the basics right now. So I'm going to skip, as I mentioned, I'll be skipping things. I'm going to skip therapeutic cloning and reproductive cloning. It's in your book. It's a very simple concept. Here it is. If you want to read about it, you can, but this is not something we're going to talk about today for time. We're going to move on to induced pluripotent stem cells, something I do want you to know about. So remember, to back up for a second, and the last lecture I told you about different types of stem cells, totipotent, pluripotent, and multipotent. And what I asked you to remember was their potency, or the power they have to become different things. So a totipotent cell can become any cell type, a pluripotent cell can become all the cell types in your body, but not your extraembryonic tissue, and a multipotent stem cell can become many things, multiple things, right? Multipotent, but not everything. And again, a way to remember that, it's the reverse alphabetical order. So TPM becomes more and more restricted in the type of a stem cell. Or if you prefer correct alphabetical order, you're getting broader. However you want to think about it, that's how you can try to remember it maybe. Toti, pluri, multi. And obviously I'm emphasizing that because it's something I want you to know. Okay, so pluripotent stem cells then are the ones that can become any kind of cell type in your body. So how exciting would that be if we could harness that power, as I just mentioned? Well, there's a few ways to get them. One way to get them is to take an embryonic body that's being discarded and to extract embryonic stem cells. As you're probably aware, there are some ethical issues and also for some people, religious issues associated with that. So that's one way to do it. Another way to do it is to induce pluripotent stem cells. So these are induced pluripotent stem cells, or iPSCs, are made to become pluripotent stem cells. They are not starting out that way. So they provide a convenient source of human embryonic stem cell-like cells. So again, to recap that, naturally your pluripotent stem cells would be in an embryo, the cells that can become anything. But because of various costs, ethical considerations, religious considerations, there is interest in other ways to create these types of stem cells. So what if you could just induce them instead of having to take them from this source? So how do you induce them? Well, let's say you take some cells from your skin, you put them in a dish, and people have done lots of tedious research, important research, long-term research to figure out what factors could you add to that media to make them become different cell types. So in other words, what was sort of on this slide here the idea that you would, I didn't go through them, but the idea that you would put different things into the media that's on these cells to make them into different types of cells. So that's, in this case, we're using different things in the media to make different types of differentiated cells, whether it's a neuron or an islet-like cluster cell. But what if you could also find the right factors here that let you go the opposite direction, right? So you're going backwards in a sense. In the first slide, I showed you you're taking a stem cell and, let's say, going to a neuron. What if instead you could go neuron to stem cell? So that's what an induced pluripotent stem cell is, or an induced stem cell is this idea that we'll take a differentiated cell, whether that's from your skin or your liver or whatever it might be, and can we figure out the formula? Can we figure out the changes you have to make in that cell to turn it back into a stem cell? How cool would that be, right? You'd have a source now of stem cells to do stuff with. So it turns out we can do this, at least in dishes. People have done a lot of research and found, for example, that you can have as few as 3 key proteins that are transcription factors that you can add that will cause these cells to become induced pluripotent stem cells. And from there, you can then try to differentiate them in a dish to these different cell types and many others. Now, there are caveats to this, and it's not working perfectly. Here are a couple of the caveats. Again, I'll be very specific. I'm not going to test you on these in this class, but it's important for you to know that there are problems with induced pluripotent stem cells that need to be overcome. And to be clear, I'm not testing you on the problems. I do want you to know what iPS cells are. Okay, so here are a couple of the problems that show up. So you'll learn about in genetics, if you haven't already, something called epigenetics, the idea that we modify our DNA in different ways. So if I tell you that these two cells have the same DNA, we know they're different because they make different RNA and protein, but what we're not covering in a cell bio class is that some of that difference also comes about, well, in fact, different RNA and protein come about because of changes to the DNA that include things like putting on methyl groups and other types of modifications to the structure. That's called epigenetics, and the two main examples are shown here, DNA methylation and modifying your histones. So the problem is that when you take a differentiated cell, a skin cell, and you turn it into a stem cell like this, you haven't, unfortunately, necessarily changed the epigenetics. In other words, you have stuff expressed to turn it back into a stem cell, but if the DNA still looks like it used to look like in the previous cell type, that can create subtle problems and not be so great for the therapy we want. Maybe the cells don't live as well, maybe they die, maybe they don't differentiate quite the way they're supposed to. Maybe they are at risk of becoming cancerous or something like that. So iPS cells tend to remember what they were because their epigenetics is what it used to be. And related to that, then, iPS cells are often full of cancer-causing mutations. If you take a skin cell from me, that skin cell is 41 years old, right? Now you turned it into a stem cell. It's a 41-year-old stem cell. It's not a 6-month-old stem cell in a fetus. So we've learned all about how as cells divide and divide and divide, you accumulate mutations, and we all are doing that. So imagine now if you're taking these stem cells from somebody who's old and has accumulated mutations, and now you're turning it into a cell that can become different cell types. This is sort of like cancer's dream come true, right? You have now the ability to divide like crazy and do lots of stuff. So it's kind of dangerous to do that, and that's that's a challenge we have to overcome as well. Okay, so that finishes up in a nutshell induced pluripotent stem cells. What I want to briefly talk about is development. And this is, how we all come to be. Remember, I started off this lecture last time with a picture of a single cell becoming, a sea urchin, and a single cell becoming a mouse. How do they get to be so different? Well, it's stem cells becoming different things. That process of becoming different things is called development. So the development is how a living thing goes from a single cell into a more complicated multicellular organism, right? So pretty straightforward concept. This is an example of a cell going through various stages to become a worm. Don't worry about it any further than that. It's just an example. Now, another term you need to know is cell differentiation. You've probably heard me mention it several times, so let's be very clear about that. Differentiation is the process of a progenitor cell, such as a stem cell, becoming a more specialized cell type. So it's how cells take on different characteristics. In development, usually this means going from less specialized to more specialized, right? A stem cell becoming a more specific type of stem cell, or a specific type of, let's say, multipotent stem cell becoming a muscle cell or something like that. And that's illustrated here in an early embryo where you've got lots of different layers forming, and then they can differentiate into cell types. And again, this is stuff that you will cover in detail when you take, let's say, a dev bio class or a higher level class after this. But just know what cell differentiation is, broadly speaking. And to put it very simply, this is happening because a cell expresses different parts of the genome compared to its progenitor cells or its sibling cells. It's exactly what we've been talking about. Cells in your body have the same DNA, more or less, minus individual mutations that may have happened. And so how do you make them different? You express different RNA and different protein. So all this slide is showing you is that you're dividing a cell here, and now maybe this one, to put it very simply, makes a neuron-specific protein from a neuron-specific RNA, and this one makes an epithelial-specific protein from an epithelial-specific RNA. Now, this is super oversimplified, right? In your actual cells, you have hundreds, if not thousands, of different things that are coordinating and changing to make this happen. Okay, so then this is development, this is differentiation. I have a fun YouTube video to show you, but I'm going to save it for the end because I want to make sure that we get through the important stuff, and then if we have time, we'll finish off with this video. Okay, so and one other thing I need you to know for this class is extrinsic versus intrinsic signaling. Now, I'll start off by saying, please don't get confused between this and extrinsic and intrinsic pathways of apoptosis. So remember, the words extrinsic and intrinsic are simply definition words in the English language. When something is extrinsic, it means it's coming from the outside, right? So from the outside of the cell in the context that we talk about. Intrinsic means something is within something else. So, something that's intrinsic to you is part of you. Intrinsic signals are coming from inside of the cell. The extrinsic pathway of apoptosis comes from the outside. The intrinsic pathway of apoptosis happens inside. So the words are the same, but they're different contexts, okay? So we talked about apoptosis, and now we're not talking about that. We're talking about signaling the extrinsic or intrinsic. Now, you already learned about this, it turns out, because, for example, when we talk about a ligand binding to a cell receptor, a GPCR, that is extrinsic signaling. When we talk about your apoptosis pathway, actually, that's intrinsic signaling, or anything that starts inside of your cell is intrinsic signaling. So you actually have learned this. It's just explicitly stated now in the context of stem cells and differentiation. So in either case, you end up doing these things we've talked about. You're going to express genes and you're going to differentiate. So let's talk about extrinsic differentiation signals. And I do want you to know what a morphogen is, okay? So I'm just making it very clear for you. You need to know what a morphogen is. So morphogens are extrinsic differentiation signals. In other words, they come from other cells. An example of this is BMP, bone morphogenetic protein. You don't have to worry about the name. So this is an example of a signal that comes from the outside, binds to a receptor, and does a bunch of stuff. Now, why is that specifically a morphogen? Because we've learned about lots of signals that come in from the outside, whether it's a mitogen or a growth factor or a survival factor. So what makes a morphogen a morphogen? It's an extrinsic signal that's going to lead to changes in gene expression that actually cause you to undergo differentiation. So to put it more simply, a morphogen stimulates differentiation. It stimulates the process of development, broadly speaking, okay? Or morphogenesis, if you want to know where this comes from. So changing. Morphogenesis is changing something. So to morph into something, right, is to change. And so that's what a morphogen is. This is a list of some of them. Don't worry about it. Just wanted to share that with you. Now here's an important point about morphogens. Morphogens are secreted from cells and have an effect on a bunch of cells, right? But so if I'm a cell and I secrete a bunch of morphogen and somebody's all the way over there, they're going to see a decreasing gradient. It's going to be highest concentration next to me because I'm secreting it, and really, really low over there, and then probably a nice gradient that's relatively even or increasing at an even slope in between. But the response to morphogens doesn't work that way. So in this example here, this blue line is a morphogen gradient. In this case, it's taking kind of a curve-like gradient. So it's high up here, it's low down here. And then this is, let's say, where it's being secreted. These cells here are secreting the morphogen in red, and it's going down in concentration as you go along. So what happens is that even though the concentration is gradually decreasing, the response to it is based on very discrete thresholds. In other words, at any given point, so this line indicates an arbitrary, well, for this diagram, arbitrary point in the distance going this way. And if you are a cell on this side and you see this much signal, you're going to do one thing, become orange, let's say. And if you see this much signal on this side, even though it's only a teeny bit less, you're going to become blue. So the signal is gradual, but the response is very discrete. It's like a binary response because the cell is looking for a threshold, and if it's over that, it's going to do this thing. If it's under that, it's going to do that thing. And the reason that pays off for the body is that, let's say you're sending a signal starting here, coming out this way. You can have a gradual signal, but the response being discrete means that you can differentiate into very discrete bone structures. Because the cells on this side know I better be a part of this bone and not whatever that is. Okay, here I better be part of this and not that. So it's a translation, right? The signal is gradual, but the translation of it is very specific to have cells become different structures as you're developing. It's a very elegant process, actually, of taking messy signaling and turning it into a very specific outcome that you need to make, for example, a well-formed limb that has all of its pieces in place and working together. So that's just a very common example of that happening. Don't worry about the stuff down here. We don't have time to get into it. So that's extrinsic signaling and how morphogens work. Any questions about that? I know I'm going a bit fast, trying to make sure you have time for review. Sorry about that, but I hope it's... Any questions? Okay. So intrinsic signaling, I think that's the last major thing I want to cover for you, although I have a few other things to talk about. Intrinsic signaling, remember, comes from inside of the cell. And this is an example of that. So this looks really complicated, but just I'll walk you through it. Let's say you've got a population down here of progenitor cells that are dividing, and this is time going in this direction, okay? So as time passes, things are happening. These progenitor cells, or stem cells in blue, are doing what they do. They're dividing, and it turns out some of them are making neurons, and these neurons are being sent over here, and they make a nice layer in your brain of neurons. What happens is, as time passes, these cells have an internal clock. One of you actually emailed me about this a few weeks back, and we talked a little bit about circadian rhythms and how cells actually have their own rhythms and their own clocks sometimes. It's pretty cool. So it turns out that after a while of doing this, after a certain number of cell divisions have happened, this cell will change what it becomes. And instead of now making this type of neuron, it's going to make instead the green type of neuron. It's going to do that for a little while, and those neurons are going to be sent up here. Then it's going to change what it makes again, and it's going to make only the red types of neurons, and they're going to be sent up here. So the same progenitor cell population over time, and time being measured in this case by number of cell divisions, is actually changing what it does. And by doing that and sending them in layers, you end up making really nice layers of your firstborn neurons and your subsequent neurons with your most recent ones up here. And then when you look in your brain, you have organized layers of neurons that can be quite useful functionally for your brain. All right, so that's kind of neat to think about. A single population, and it's not, I'm simplifying a bit here, but a single population of stem cells is able to really carefully lay out the order of various neurons. So that's intrinsic signaling, because this was cell cycle dependent changes in gene expression that happen inside of the cell, independent, inside of these stem cells, independent of anything outside. That's why it's intrinsic signaling, okay? Let's skip this slide for time. Although it's kind of neat, you can look at it on your own. So the big picture takeaway so far then is that differentiation signals control the direction of cell development. You can think about stem cells as having a fork in the road, and they have to decide, do I want to be a muscle cell? Do I want to stay a stem cell? Do I want to be something else? And that differentiation is controlled by the signals I just described to you, whether they're extrinsic or intrinsic. This is a diagram to the right side here of cell lineage. These are terms I'm not going to test you on: cell lineage, cell fates, and cell fate specifiers. In other words, I'm not going to specifically test you on them, but guaranteed you have to know this stuff for your entrance exams. It's actually fairly self-explanatory, so I encourage you to study this. This is a good study slide for your entrance exams, where you start with a hematopoietic stem cell, which becomes a progenitor cell, and then you have other cell types coming from it. You don't have to worry about the abbreviations, but looking at this helps realize how you make different categories of cells in your blood, including immune system cells and others. And so these are all different cell fates that are coming from a given cell lineage. A cell lineage just means this is how you get here. Just like you have your own lineage, right? Your parents, your grandparents, and so on. So that's what a cell lineage means. And cell fates are the end fates you get to. And then these signals I told you about are cell fate specifiers. Again, I'm not going to test you on these three terms, but if you don't learn them, you're doing yourself a disservice, so I encourage you to think about those terms and kind of absorb them because you're going to need them. Okay. Stem cell therapies are a fun topic. One of the questions I often get is, are stem cell therapies on the horizon? And I often say, guess what? They already are in use.
Bone marrow transplants, which have been going on for decades, are a form of stem cell therapy. That's probably the most common one that you're familiar with. So what happens when you have leukemia, for example, is you have a cancer of your blood and bone marrow, and to treat it, you have to kill everything that's there, the cancer cells, your regular cells, your stem cells, and start over. And now what do? Because without those stem cells, you're going to have a hard time having a proper, basically, blood supply that has everything you need, the right cell types in there. So what you can do is find a donor who matches you and put in hematopoietic stem cells from the donor, replace that population. They will actually take hold in that niche. They'll hang out there and they'll start to proliferate, and they'll make the different cell types that you need shown here again. So stem cell therapies are already there. Obviously, as time passes, we're coming up with more and more exciting and interesting types of therapies as well. One example of the progress we're making, these are mouse eggs made from skin cells in a dish. This was a paper in 2016. So let me pull up this. So there is a lot of interest in being able to create organisms and therapeutic tissues right from stem cells. And so this was both exciting and created worry about the ethics of, let's say, doing something like this for humans, because you could now make human eggs using the same technique, in theory. I mean, there's differences and it's not that easy, but what was exciting here is that a group basically took skin cells from mice, made them into eggs, so your very core type of a germ cell, and then used those to actually make these 11-month-old mice from eggs that were taken from a dish. So these are mice made from induced stem cells, induced pluripotent stem cells. That's pretty wild, right? And you have the links if you want to go through and read about this. It's really quite remarkable. Another example, you've heard already about CRISPR-Cas9. Just last year, 2018, a team at Gladstone Institutes in the Bay Area, they took mouse skin cells and turned them into, again, iPS cells, so the same thing I just told you about, but they now did this using CRISPR-Cas9 technology by activating a single specific gene. And so now you're talking about really specific types of activation that you could do with a new technique, and that could get around some of the worries that you have using traditional techniques about what else you're changing. Of course, every new technique has its own worries, and as Srivatsan told you, CRISPR-Cas9 has its own concerns, such as off-target effects, unknown consequences of what we are changing, as well as, of course, ethical considerations about what we should actually be doing in humans. But that's also here for you to read if you're interested. So I think I'm doing pretty good on time, so I'll pause and ask any questions about anything I've just covered. So you guys are sleepy today, busy study. Yes?
For the extrinsic pathway, you said that they were discrete and gradual, right?
So, yeah, for the extrinsic pathways.
They were discrete and gradual?
So the question is, for the extrinsic pathway, I mentioned there was discrete and there was gradual. So the question, I guess, is what's what? Right, so the gradient is gradual, right? If I took marbles and I tossed them here, they would roll away, and I'd have the highest concentration of marbles here and the lowest over there. And if you looked at it, it'd probably be a gradual change, right? So that's your gradual gradient. But what's discrete is the response. So if I had, and I'm just, I don't know, if you've got those hungry hippos that eat the marbles, and one hippo is sitting here, and one's over here, and one's over here, their response to the number of marbles doesn't have to be gradual. It can be discrete. Maybe this one knows as soon as I eat 3, I'm going to do something. This one knows as soon as I eat 2, I'm going to do something else. And they don't care if there's 4 or 1 or 5,000 of them. They're going to do what they need to do once they eat the number of marbles they have to eat. So the gradient is gradual, but the response is very specific and discrete. Does that help? Okay. Mm-hmm. Nothing like a hungry hippos analogy, right? Is there another hand? Yeah. Sorry, I can't hear you. Oh, the backs of their, yeah, I'm not sure, to be honest with you. That could be some sort of pattern just when they were born. It's also possible, in fact, likely that they had their hair shaved because somebody put a probe on there to check something. But yeah, I don't really know. Yeah.
The CRISPR-Cas9 induced iPS cells equal or more possible to cause mutations?
Then which ones? Yeah, that's a good question. So the question is, when you make an iPS cell, induced stem cell with CRISPR-Cas9, is that more or less likely to cause mutations in those stem cells than if you had made those iPS cells using the more traditional techniques, like what I told you about 3 transcription factors that were introduced and all that kind of stuff. The short answer is I don't know, and I don't think people really know. The longer answer is that you can make an argument that it's more likely to cause mutations because if you are making induced pluripotent stem cells by, let's say, hypothetically adding 3 proteins in, you can make an argument you haven't changed the DNA, so perhaps you're not inducing mutations, but the counter-argument is those proteins could be doing something that could lead to mutations. So, and with CRISPR-Cas9, we know that we have the risk of off-target effects. The other side of the argument is that with CRISPR-Cas9, you're making a specific change, and if you can show that your off-target effects aren't there, and there are ways to try to do that, then you're actually more specific because you're not just dumping a bunch of proteins on something and not knowing what's going to happen. You made your one change, it did the effect, and it's going to last because it's a genetic change. So there's really, it's not clear, but there's arguments on both sides of that.
So for the bone marrow therapy, for the leukemia, so if you like kill every cells from like chemotherapy, so does the person die?
So the question is about the therapy for leukemia. If you kill all the bone marrow cells, would the person die? If you don't do an intervention, it's a huge risk, yes. At the very least, they'll be incredibly ill, and they are likely to then die from that. So when you do any type of chemotherapy for any kind of cancer, you're always trying to strike this really difficult balance between killing the cancer and not killing the patient. And there are cases, in fact, where patients will die of side effects from chemotherapy, but if you're terminally ill from cancer, it's oftentimes worth that risk. In this case, yeah, you have that problem, and that's why you have to find a source of putting those stem cells back in there. You can also hope some of the stem cells survive on their own in the patient, but that's not always going to happen. Yes?
I actually sent you that circadian rhythm.
Oh, yeah, that's you. Okay, Paige then, right? Yeah. Yes, okay. Good to put the face with the name, yeah. On the last lecture.
So, I was wondering with the intrinsic signaling, if that's completely only time dependent and not dependent on concentration. I mean, not concentration, on the gradient of the morphogen, or if it involves.
Yeah, so the question is, oh, yeah, go ahead.
But if it involves any sort of gradients.
So the question is the intrinsic signaling truly independent completely of the gradient that might be outside? Yeah, so that's for the purposes of this class, yes. In reality, the biology is much more complicated than that. In reality, you'll have an intrinsic mechanism that is independent of the outside in terms of what I'm telling you, yes. But that cell is being bombarded by all sorts of signals, and it's quite likely that there are signals coming in that will also influence the same pathways. But that's sort of teaching you what is intrinsic and extrinsic signaling as units. But it's very true, as we've seen throughout class, that different things we teach you, whether it's the pathway on RAS versus a pathway on apoptosis or something else, things are always interacting with each other. So, yeah. Yes?
Does this also depend on telomeres? How many times the one cell will divide to differentiate in one type?
Does it? Yeah. So the question is, does it depend on telomeres? Yes. Well, in the long term, yes. As cells divide, their telomeres get shortened, and eventually that will affect the cells as they get older in terms of aging, and the cell may not be able to. Will have to apoptose most likely. So it can, yes. But in the short term, when you're talking about signaling here, based on cell cycle to make a change, like let me make more neurons, you're talking about young cells that are stem cells, and so that's not really going to be an issue until the individual is much older. Yeah. Okay, so let me wrap up here. Very quickly, just to mention that human cloning isn't happening. I say this with an asterisk because in some countries across the world, we don't have as good regulation, and there have been rumors for some time and about things that might be happening, but let's hope that, anyway. It's just, but at least in the United States, we're not doing this for obvious scientific and ethical reasons. Animal cloning is very common, and that's all I'll mention about that. A couple of fun things to end with. Cloning can lead to all sorts of interesting things. People are cloning their pets now, getting their cats cloned. This one shut down because not enough people were buying expensive cloned cats. The company had to reduce its price from 50 grand per cat to 32K because, that's affordable. And one of my favorites is that you can go and clone your dog in Korea. It used to be $100,000. Now it's only $50,000 if you've got a dog you want to clone. Takes about 6 months to a year. So there is a lot of cloning going on. And I want to finish by mentioning a brief rant about GMO issue as part of my general comment about science versus what's called pseudoscience, which I think is poorly named because pseudoscience still has the word science in it, and that kind of gives it more credibility than it should. So what I want to just say to you is this. It's going to sound a bit cheesy, but you guys are the next generation of leaders in this country, right? You're going to be scientists, physicians, dentists, nurses, et cetera, and scientists in some way. So I would encourage you to try and make a difference and do your own research at reputable websites and taking a fact-based approach, which is so important these days, given all the things you find on the internet, in terms of making decisions and educating people around you. So for example, now you'll find plenty of foods that say, oh, it's GMO-free, as if genetically modified foods are such a bad thing. They're not. They're not. We've actually been cloning foods for thousands of years, and we've been genetically modifying crops in the United States for decades. It's just that it became an issue more recently because folks decided to make it an issue. Now, why this gets to me a bit is because there are countries in this world where food is not as available, and being able to give people things like a genetically modified rice strain that is more resistant to, let's say, a pest eating it, instead of having to put on nasty pesticides, helps those farmers produce more food for their populations at a lower cost. And when we have an anti-GMO movement, which in a developed country people can get away with because most of us are able to buy food, it might just affect for you what you buy on the shelf and its cost, but it has very dramatic effects in poor countries for the access that people have to food. So that just kind of gets to me a bit. It's like, okay, it's very easy to be anti-GMO when you're in a developed country and not even think about it, but it has huge ramifications for developing countries where people do need access to better, safer sources of food. So something to think about when you automatically take a stand, is it always true? Now, just like with anything else, if I create a mutation, I could become a superhero or a supervillain. And if you're mutating a food, you can benefit, or if it's done in a poorly regulated, dangerous manner, it could also be bad for you. In other words, just like anything else, there's both good and bad aspects to these things. And it's really important to take a subtle, nuanced, scientific view of these issues rather than just deciding I'm going to be pro or anti something because somebody online said it was a good idea. So I encourage you guys to look into that, whether it's looking at genetically modified foods, or here are some more comical but scary examples. This is from last year, a food safety expert warning about the latest bizarre trend in Silicon Valley of $60 raw water, where people are actually going to like, I don't know, ditches and grabbing untreated, unfiltered water and drinking it because they think it's better for you. Okay? People are actually doing this. This is nuts. You can get not just hurt, but you could die from something like this. And why does this happen? Because somebody gets online and says, hey, raw water is better for you, so let's do it. You may have heard about the food babe, as she calls herself. This is one of her quotes. If you can't pronounce it, that probably means the material has been part of the human diet for a minute period of time in terms of the human evolutionary or developmental process, which translates to, if you can't say it, you shouldn't eat it, because that makes sense. So, if I can't pronounce hydrogen dioxide, then I shouldn't be drinking water. All right? And this is a little bit more about that. For example, if she finds an ingredient, an ugly-sounding chemical, propylene glycol, which she said was in beer, not true, and finds a secondary industrial use, and then turns it into an issue. In this case, she confused her chemicals because the ingredient in beer is actually propylene glycol alginate, which despite its name is not close to this thing and is not antifreeze. It actually comes from plants. So it's a really good idea to do your own research, and not just that too, but to educate people around you to go and find the facts, not to just see what you read on a blog and go with it. So that was my brief rant about the importance of science and logic. 11:35, so I will not play the whole video for you, but I'll quickly just play part of it. So remember I told you about how things develop, differentiation, and how it can be different, the sea urchin and the mouse. There's a whole field called Evo-Devo that looks at how the evolution of the developmental processes. And this guy does some very nice parodies. And I'll just play like maybe the first minute since we're out of time. Maybe you guys have seen this before. It's one of my fun things to do in the last class. So. Uh-oh. First minute.
1,000 fates. Did you ever figure how they know? Be mad. We are built of modules combined in a planned out way. Each new piece must be told where to go. Oh, now there's a science helping us to understand how our cells encode this architectural plan, signaling each other with genetic tools. Oh yeah, wow. Phenotype the interface for mouse and man, genotype the files and the subprograms. What then are the switches, circuit boards, and root code? Evo Devo, looking at the logic in the ways that we grow. Every gene directed by a signal key code. Proteins that can activate, enhance, or veto. Evo Devo, the signals are controlled by other genes that signal, calculating in a network labyrinth though, where the heart and liver and the hands and feet go. Signal mapping till these region what it ought to be, yo. With circuits so deep within the core, they're older than the millions are waking up, baby. In a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes. As the cyclonic shield, see down they cascade like a domino, like you and I, Drosophila. The path that makes us optical was laid a long, long time ago. Back before we blew up the Cambrian, the K-bomb bomb. Now my eye protein can make you see out of your bomb bomb. And hedgehog and its relatives like Indian and Sonic. Set up in a gradient of segment embryonic. Split four pins and asymmetric parts up in the planet. Flipping on genetic switches and logic from Devo to Evo, adult and embryo. Mostly don't evolve in the genes of the genome. Save for the mutation in the regulation. Keep the building blocks and swap their activation. From Devo to Evo, parts of alter egos. Homologs evolve from repeats in the schema. Switch a couple bases in the proper places. You'll be watching flies go legs out of their faces. Oh yeah, Evo Devo.
All right, so I'll stop it there for time. I know. But I want to make sure you guys get to review your exam. So you can find this online and watch the rest of it. I'm as sad as you are. It's also really hard for me not to start dancing to this, which you do not want to see. So, all right, you want to connect up? Yeah.
And while he sets up, a couple of quick reminders. Your exam is on Thursday. I hope you guys all know that. B, I'll be having extra office hours right after this class, so come see me upstairs if you want further help, and your TAs as well. And then C, so we've hit 70% for the eval rate, so congrats, because we still have until Sunday. So we'll add those 2 points to everybody's extra credit total, maybe later today or something, on Blackboard, so you'll have that available. And then I am going to this evening add a running total of your points so far, just so you have it. It's not anything remarkable. It's the same thing as if you added up all your exam scores plus your extra credit points. But for those who want me to do that for you, I will add that column and make it visible. But please note that that column does not account for dropping an exam, so you'll have to kind of figure that out yourself. All right, and I'll send an email this evening with that info and a bunch of other stuff as we get close to the exam. All right, so Joe's going to go ahead and take you through the first half of the semester review. And then again, Dr. Linner and I are both here to answer any questions.
Okay, can everyone hear me okay? All right, hopefully it's loud enough. I'll try to speak loud. If you guys can't hear me, just raise your hand and let me know. I'm going to go through the material for units 1 and 2 pretty briefly. If you guys have any questions, feel free to raise your hand or have to flag me down, and we can stop to talk about for a second. But otherwise, we'll be giving a pretty kind of quick and brief overview, and then Austin will do units 3 and 4. So one of the first things that we talked about and discussed in this class was the similarities and differences between different cell types. So here I have a picture essentially of a prokaryotic cell, in this case a bacterium, and also eukaryotic cells. These are electron micrographs. Some of the things you might notice is that the similarities are things like both these cells have a cytoplasm, both have ribosomes, and both have DNA in them. However, there's also lots of differences. So for example, the prokaryotic cell doesn't have a nucleus to actually house this DNA. And additionally, the eukaryotic cell has lots of these membrane-bound compartments called organelles. And these can actually house and kind of compartmentalize different processes inside the cell. And so a lot of them are listed here, and you guys can definitely refresh yourself and study them as well, and make sure you know them for the exam. So these are like kind of some of the similarities, the differences between prokaryotic and eukaryotic cells. Another kind of idea we talked about was plant versus animal cells. These are both eukaryotic. Here's kind of a schematic design of both these types of cells, and again, showing a lot of the organelles. One thing that's kind of interesting, for example, is that animal cells, they have a centrosome, which essentially enables them to kind of organize and create cytoarchitectural structures. What's interesting is that plant cells actually don't have a centrosome. Instead, they have cell walls and a vacuole, which kind of serves a similar or related purpose. Additionally, too, plant cells also have chloroplasts where they can essentially do photosynthesis. And so animal cells do not have this organelle. Everyone good so far? Okay. So one of the next topics we cover is a bit of like chemistry and kind of and things like that. So I'm going to briefly go over just a couple things from the chemistry section. So one is talking about covalent bonds. So a covalent bond essentially is when there are electrons that are being shared between two atomic nuclei. And in this case, it could be a nonpolar covalent bond when these electrons are shared equally across the atomic nuclei. But additionally, there's also polar covalent bonds. For example, this water molecule here, where the oxygen atomic nuclei kind of hogs these electrons and unevenly shares them. And this kind of polar uneven sharing of these electrons actually causes a partial negative charge near the oxygen atom and a slight positive charge near the hydrogen atoms. And so this kind of polar covalent bond actually creates a partial charge distribution across the water molecule. So one of the reasons this is so important is that this actually allows hydrogen bonding, which is a type of non-covalent interaction. So a hydrogen bond is when a partial charge on one molecule actually interacts with a partial charge on another molecule. And so this example, it's two water molecules forming a hydrogen bond. Now, another thing I want to briefly go over is also ionic bonds. So instead of covalent bonds where electrons are being shared, in an ionic bond, one atomic nuclei actually gives up an entire electron and donates it to another nuclei. And so it creates a full charge difference between these two nuclei, which allows them to bond to each other and to attract each other. Additionally, you might want to review van der Waals interactions as well, but again, for time, I'm going to kind of skip this, and you guys can review it on your own. So another topic that we covered essentially are macromolecules that are important for cells. So in this kind of schematic, one of the important things to take away is that macromolecules like polysaccharides, proteins, and nucleic acids are some of the structures that are needed within these cells, and they're made up of different subunits, which are like monomers of sugars, amino acids, and nucleotides. And the way that these macromolecules are made, they're made in such a way of the different synthesis reactions. So again, these macromolecules are polymers made up of these monomers over here. So the way that the synthesis reaction works is one specific type of reaction you need to know is a condensation reaction. And so a condensation reaction is when an OH from one of the subunits and a hydrogen from the growing polymer actually combine to create H2O, a molecule of water. And in this way, the subunits actually joined to the growing polymer. And kind of the opposite of this is a hydrolysis reaction where you can actually split individual subunits away from the polymer by adding a water molecule. Everyone good on that so far? Awesome. So the next topic that we covered was on energy. And so this is, I think, is a really great review slide for you guys to kind of think about the different modes or different types of energy and how they're used in cells. I'm going to go through it really briefly, but for example, A talks about how energy can be stored essentially by the position of different molecules or different items. So here it's talking about the position of a brick and how there's potential energy stored in its position. As far as an analog for cell biology, you guys should think about things like concentration gradients, where energy is actually stored by having, for example, protons or other molecules in a concentration gradient where on one side of a membrane you have a lot more, on the other side you have a lot less. B talks about energy that's stored in kind of vibrational energy or heat energy in the cell. C also talks about chemical energy that's stored, and a really good example of that is essentially electrically active cells like neurons that actually set up a voltage difference across the membrane. And kind of almost similar to this kind of potential positional energy that's stored, the electrical energy, like voltage across the membrane, can also be a way to store energy. And D lastly talks about electromagnetic energy, so obviously photosynthesis and essentially converting electromagnetic or light energy into chemical energy is a very important part for any photosynthetic organism. So if you're talking about cellular energy, it's also really important to go over the idea of activated carriers. So the way I like to think about this is that activated carriers are usually small molecules like ATP or NADH. And the way that they're used is that they're like an energy currency for the cell. They're available to use right away, and they contain a relatively speaking small amount of energy. And the way this works is that in catabolic reactions, you have food molecules that have high energy bonds. those molecules and those bonds are broken down through energetically favorable reactions. And what happens is the energy that's stored in those bonds actually gets moved to activated carriers. What happens after that is the activated carriers can actually drive energetically unfavorable reactions, such as like synthesizing large molecules or building different components of the cell. And what's important about that is that these energetically unfavorable reactions wouldn't happen without the energy input from these activated carriers. And what's really great is that this diagram shows that it's a cycle, because after these activated carriers run these anabolic reactions, they're actually free, and they're in an inactive state and are free to pick up more energy from other catabolic reactions. Is everyone good on that so far? So the last thing to think about as far as energy in terms of cells is actually enzymes. So an enzyme in a cell is essentially, it's a biological catalyst. And so one thing that's really important to know is that in cell biology, almost all enzymes are proteins, and the only exception is that a few enzymes are also RNAs. And it's important to remember, and that's what it's important to remember this diagram on the right here that kind of explains that when you have an enzyme, you're not changing the amount of energy that's stored in your reactants or your products of a given chemical reaction. Instead, what you're doing is you're lowering, the job of an enzyme is to lower the activation energy that's required to kind of drive this reaction. So you can think about an enzyme as lowering the amount of energy you have to invest to get the chemical reaction kick-started. That's the way I like to think about it. So you might ask, why are enzymes even important? So enzymes are actually really critical for regulating different biochemical pathways. So on the bottom here, it's a simple linear schematic of like an example biological pathway. And the reason this is important is that, for example, molecule A is catalyzed by enzyme 1, turns into molecule B. So in this case, B is kind of like the product of that reaction. But B is actually an intermediate because it's used as the reactant in the next reaction, which is catalyzed by a different enzyme to turn into C. So in this way, you have a biological pathway where each kind of product of one reaction is the reactant for the next. So again, in this kind of example, molecule F would be the final product, and for example, B through E would be intermediates. This diagram on the right just kind of shows what I've already talked about with activated carriers, energetically favorable and unfavorable reactions. You can think of the blue cog as an energetically favorable reaction. It's putting energy into this orange gear, an activated carrier, which is helping to drive this red cog, which could, for example, be an energetically unfavorable reaction. So in this way, biochemical pathways can be linked together to help drive one another. The next big idea we talked about was protein structure. So really briefly, I'm just going to go over the 4 types of protein structures. This slide is kind of has a lot of information on it, but I think focusing on this bottom part here will help you remember that the primary protein structure, is simply the sequence of amino acids that are strung together. So again, a polypeptide or a protein is a polymer made up of amino acid subunits. The primary structure is just simply that order those amino acids. There's also higher order protein structures as well. So for the secondary structure, it's types of structures like alpha helices and beta sheets that come about from hydrogen bonding between the backbone functional groups. And so these are not the R groups of the amino acids, but they're backbone groups. Tertiary structure accounts for like the 3-dimensional folding of a protein inside the cell. And this actually results from a couple different things, including hydrophobic effect, electrostatic interactions, and disulfide bonds. It also comes about from hydrogen bonding between the R groups of the amino acids. And then lastly, there's also the quaternary structure, which is essentially when 2 or more subunits of a protein come together and interact. And so not all proteins are going to have a quaternary structure, but if there are subunits to a protein complex, that's what that accounts for. So the reason I'm telling you about these different types of structures is that the primary through quaternary structures of proteins account for its 3-dimensional shape. In the 3-dimensional shape of proteins, it's really important that it's very important for protein-ligand interactions. So one of the big concepts we covered was that a ligand, essentially it's a molecule or ion that binds reversibly to another molecule, can specifically interact with certain proteins because their shapes are compatible. In this diagram, you can see these like purple non-covalent bonds. These are all able to form, and the 2 molecules are able to stabilize each other because their shapes fit into each other very specifically. Down here, you can see a couple of real-world examples. For example, hormone receptor binding to its ligand, and also antibody-antigen interactions, which are another example of these like protein-ligand interactions. Does anyone? I went through that super fast, and that's all of Unit 1. Does anyone have any questions? Anything I talked too fast on? Anything you guys are confused about? All right. So next, I'm going to go over Unit 2 pretty briefly. So with Unit 2, we started basically with a discussion about lipid bilayers and different proteins that are embedded in them. So I think this is kind of a pretty good review schematic to kind of illustrate that. So here we see the lipid bilayer, the plasma membrane. And we actually see different proteins that are a part of it. So you can have integral proteins that actually span both sides of the plasma membrane. You can have lipid-linked proteins where they're linked to one side of it. You can also have peripheral proteins that associate or are connected to other proteins that are embedded in the plasma membrane. Another key idea we talked about was how proteins that are embedded in these membranes, how are they free to move and how are they restricted? So this schematic here on the left was from one experiment that showed that under kind of default circumstances, different membrane proteins can actually laterally diffuse throughout the membrane. And so you can kind of think of this as the default state is that if there's nothing anchoring or blocking different proteins, they are more or less generally able to diffuse throughout the plasma membrane. On the right is actually an example of one way that the movement of proteins can be restricted. So here is actually the cell cortex. So this is like an example of scaffolding or cytoarchitecture that is just underneath the plasma membrane. And this is an example of how different proteins can actually be anchored or attached to the scaffolding, which keeps them locked in one place and restricts their movement. When you guys are reviewing for the exam, it might also be important to look at tight junctions. I don't have a slide for that up today, but you can also review how tight junctions also might restrict the movement of some proteins as well. So next, another thing we discussed was the major classes of membrane-bound proteins. So I just discussed anchoring proteins to you guys. So I also want to cover transporters and also really briefly receptors and enzymes. So for transmembrane transport, it's important to remember that some molecules can actually cross the lipid bilayer, the plasma membrane, pretty readily. These tend to be small, nonpolar molecules like gases and also steroid hormones you guys learned about. As you go kind of further down this list, ions and things like that definitely need help to cross the plasma membrane. And so we'll be talking about different transporters and channels that allow them to do that. So this is a really important slide talking about the energistics of how, like what kind of energy is required to actually move these molecules around in a cell. So what you'll notice is that channels are always involved in passive transport. And what the passive transport means is that a transport of a molecule across the membrane is passive if it doesn't require the input of energy. And so in this way, passive transport allows molecules to move with or down their concentration gradient. Additionally, some transporters are also passive and also move molecules down their concentration gradient or with their concentration gradient. However, some transporters are also active. And so what this means is that with the input of energy, you can actually pump or move different molecules against their concentration gradient. And so that's what this diagram is kind of explaining here. Any questions on any of that so far? Question? Yep.
Coming in the cell for the signaling is the passive transport, right?
Sorry, can you say that one more time?
The ligand coming in the cell
Mm-hmm.
is the passive transport, right?
So yeah, so for cell signaling, for example, hormone receptors, the ligand. So the question was, for cell signaling, do you have ligands that actually come into the cell, and is that passive transport? So the answer is yes, sometimes. So a lot of extracellular signaling is when a ligand binds to a receptor that's on the cell surface. There is, however, examples of hormone signaling where the hormones are able to pass through, they're able to diffuse, actually, in a lot of cases, through the plasma membrane. And because of that, the receptor for that ligand is actually located in the cytoplasm instead. So in the case of like hormones or like ligands binding to hormone receptors, yes, it would be like passive diffusion through the cell membrane. Any other questions on transport? Awesome, okay. So a really big chunk of Unit 2 was actually on cellular respiration. So I don't have time to go into all the details, but this is a very good overview slide that breaks down the stages of cellular respiration. So to go through it, so the stage 1 of cellular respiration is taking like food molecules and breaking them into simpler subunits. For stage 2, these subunits are actually moved into the cytoplasm of the cell, and the cytoplasm is where most of stage 2 takes place. Essentially, the end goal of like the second step in cellular respiration is to make acetyl-CoA that can essentially drive the citric acid cycle in stage 3. One important part of stage 2 is that glucose is actually turned into pyruvate through glycolysis. And glycolysis produces some small amounts of ATP and also NADH. And again, at the end of the stage 2, that pyruvate from glycolysis, along with fatty acids and amino acids, is then turned into acetyl-CoA. For the third stage of respiration, this happens inside the mitochondria. And a large portion of this is essentially acetyl-CoA, which is being introduced to the citric acid cycle. And this produces CO2 as well as NADH. And so again, NADH is one of those activated carriers that I just reviewed with you guys. The second part, or actually the second part, so to speak, of stage 3 is oxidative phosphorylation. And so this is the process where NADH and other activated carriers actually help to produce lots of ATP through essentially the electron transport chain and oxidative phosphorylation. So I have a separate slide for that in a second. The super big picture is think about cellular respiration as the complete oxidation of fuel molecules. And again, this is kind of the overall chemical reaction right here. One kind of caveat to this process is actually fermentation. So fermentation is the partial oxidation of glucose. And whether it's taking place inside of a muscle cell or inside of a yeast cell, the main idea here is that glucose, through glycolysis, gets turned into pyruvate, but instead of that pyruvate being turned into acetyl-CoA, instead it's turned into either lactate, lactic acid, or ethanol. And the purpose of this is that these processes can happen in an anaerobic environment when there's no O2 available. And specifically, the purpose of this reaction here, turn the pyruvate to the lactate or to the ethanol, the purpose of that is to regenerate the NAD+, which then can be used to keep running glycolysis. So then to go into a tiny bit more detail about oxidative phosphorylation, this is kind of one of the last steps in cellular respiration. The main idea here is that NADH that's produced in the citric acid cycle is used to actually create a proton gradient that is in the inner membrane space of these mitochondria. And so this NADH can actually take, as an activated carrier, its energy can be used to pump protons, such as hydrogen ions, in this inner membrane space. And then these ions can then flow down their concentration gradient through ATP synthase to actually generate ATP. Any questions on that so far? It's a lot to cover, but it's a huge chunk of Unit 2. Okay, briefly, I want to talk about photosynthesis. So photosynthesis can be kind of broken down into two separate steps. So you have the light reactions in stage 1, you have the dark reactions. What's important to remember is that the dark reactions are light independent. So they're not inhibited by light, but they can take place whether or not the plant or the organism is experiencing or any light or not. So again, the super overall big picture here, you can see the summary chemical reaction here, is that in the presence of light, essentially H2O, the water can be used to create oxygen and also generate ATP and NADPH for the plant cells using like the thylakoid membrane. For the dark reactions, this ATP and NADPH can be combined with CO2 to essentially do the carbon fixation cycle. And so in this way, the plant cells are actually going to generate sugars, amino acids, and fatty acids. Any questions on that? Okay. Another topic, and probably one of the last big topics we talked about for Unit 2, essentially is protein trafficking. So we just talked about all these complex cycles where energy is being made, sugars are being assembled, things like that. But all these cellular pathways take a lot of proteins and structures to be in the correct place at the correct time. So how does that happen? So one thing to keep in mind, one thing we talked about was essentially signal sequences. So these are stretches essentially on a protein that can either be at the N terminal, the C terminal, or they can also be internal. They're going to direct the protein where to go in the cell. And so here we can actually see a schematic of different locations in different areas. The signal sequences can direct these proteins too. So for example, we talked about nuclear trafficking. We talked about different signal sequences transporting proteins across membranes like the mitochondria and the ER. And then lastly, we touched on trafficking signals actually move proteins into vesicles so they can either be secreted outside the cell or actually be integrated into the plasma membrane. So this is a brief kind of summary about exocytosis and endocytosis. So the takeaway here is that if you have a protein that's either soluble, it needs to be secreted, so it needs to be pushed outside the cell into the extracellular space, or it needs to be inserted to the plasma membrane. This is going to be done through essentially the protein being trafficked from the ER to the Golgi apparatus. There it can be modified, and then it's actually transported in transport vesicles that are going to fuse to the plasma membrane. And so this is kind of like the basic mechanism about how you get soluble proteins to be either secreted from the cell or proteins to be inserted into the plasma membrane. Additionally, we went over a little bit of endocytosis as well. So here's a slide that's on clathrin-mediated endocytosis. And essentially, this is just kind of like a mechanistic slide that tells you how does this happen. So briefly, essentially, you can imagine this as the plasma membrane. And there are cargo receptors that bind cargo molecules. So this kind of receptor-ligand interaction you guys have learned about. What happens is the binding of the ligand to these receptors recruits adaptin, which is like a type of adapter protein, which will bind to the receptor. And then clathrin, kind of this curved protein, will then bind to the adapters. As this process happens, the curved clathrin proteins will actually cause this vesicle to bud inward from the plasma membrane. After it buds quite a bit, there's another player, another protein called dynamin, which will actually cause this vesicle to be cut away from the plasma membrane. And essentially, this vesicle can then float away from the plasma membrane as kind of endocytosis has been mostly complete. The last step essentially is just that the uncoating of these different adapters and clathrin proteins, and then this vesicle containing whatever cargo, containing any kind of proteins or signals or ligands that was outside the cell, can be trafficked anywhere into the inside of the cell to kind of get those molecules and ligands where they need to be. Any questions on anything from Unit 2? That's it. Any more questions? All right, awesome. I will hand it over to Austin, and he'll go through Units 3 and 4.
Pocket. And how do clips work? Hello. Can you all hear me? Does that work? Is this nice? Okay, cool. All right, so we'll just get right into it with exam 3. So the slide just talks about some of the stuff that we're going to go, or the things that we're going to go over in this snippet of review. So I'll just move on to the next slide. So for here, this is just talking about, there we go, upstream versus downstream. Remember Dr. Saxena was talking about the flow of a river where things upstream happen before the things that are downstream. So like A goes into B, goes into C. And this is very important because most things that we're going to talk about from here on out involve some type of cell signaling, and upstream and downstream is how cell signaling is just referred to pretty much in all facets of biology. The arrows here show, oh yeah. Hello, yeah? Okay. The arrows here signify activation, so A is activating B, which then B is activating C, and here we have upside down T's, or pretty much just any T that you see in general means inhibiting. So if there was a T between A and B, it would mean that A is inhibiting B. And so for here, a lot of cell signaling, most signals can't just go into cells as they so freely choose. Oftentimes there will be a receptor on the outside of the cell where a signal molecule will latch onto, so these would be like a ligand, and you will have, all this could be considered downstream from that, but signaling throughout the cell, which you can get a varied amount of responses from, depending on what type of signal the cell is receiving and what type of cell is actually receiving that signal. Here you can see positive and negative feedback. So for positive feedback, you can see that X is making, or X promotes Y, and there's a little arrow going back here showing that Y will then promote X to say, hey, you're doing a good job, keep making more of me. But in negative feedback, as you see the T structure here, the little T symbol here shows that Y is inhibiting X. So in that case, Y would say, all right, there's enough of me right now. We really don't need any more of me. I'm the greatest there is. Stop, please. And so phosphorylation is one of the very important things with cell signaling, because oftentimes phosphorylation is what will activate or deactivate something. And so here you can see with the use of ATP and ADP, or the use of ATP phosphorylating something, that will then turn something on, which you'll see signaling here. And then if you have a protein phosphatase, which will take that phosphate group off, that turns whatever protein that was on off. Or there is GDP and GTP binding, where if you have something off bound with GDP, some type of protein will knock off that GDP, and now GTP can bind, which will help the protein signal. And then a hydrolysis, which, similar to like a phosphatase, will take a phosphate group off that GTP now, and it looks like it used to, with the GDP and off protein. Any questions so far? Okay. And so cells can receive signals in a couple of different ways. The first here is an ion channel coupled receptor, where you have signal molecules binding to a receptor, and that opens up a channel for ions to flow through, similar to what Joe was talking about earlier. Then there are G protein coupled receptors, where you have a signal binding to a receptor, and that will activate a G protein, which then will activate downstream effects. The heterotrimeric G protein, where you have G alpha, beta, and gamma all bundled together, is an example of a G protein. And then there are also enzyme coupled receptors, where you have two separate inactive domains that, once they receive a signal, will come together and normally dimerize. RTK is a good example of this, where you have, when they dimerize, they start to phosphorylate each other and create activation. Or you will have a signal molecule that won't function unless it has two parts that come together, but they are very similar. And so this is that heterotrimeric G protein that I was talking about, where you have your G alpha with GDP bound to it. Once your receptor protein gets a signal, it signals to that heterotrimeric G protein, and GTP pops off, GTP goes on, the alpha and beta gamma compartments separate, and then they both can do their own signaling jobs. And so if you remember a little earlier, I had mentioned that depending on the cell that you are, receiving a signal will have a different effect. Adrenaline is something that can do that in multiple cells. So here you can see that the end result of receiving the adrenaline signal is glycogen breakdown, which happens in skeletal muscle, but depending on if you're in the heart, or if you're a heart cell or a fat, then different things will happen, like the increased heart rate. And so this is also a very good pathway to look at and practice for what would happen if something went wrong in this pathway. So let's say that your receptor couldn't receive adrenaline, then all of this wouldn't happen. But if there's a different example would be like, for some reason, if you couldn't activate pKa, then the beginning section with this whole G protein signaling could happen, but you still wouldn't see the final downstream effects. And so it's also very important. All of this is going to be very important because it's the review slides. But as you can see here, you have signaling to an RTK, which will activate it. And the highlight of this slide is that you get this GT pace RAS by activating it, basically, by taking GDP off and putting GDP on. But the main part is this GT pace, is that it basically takes off a phosphate group from the GTP, making it GDP. And it's very important that there are proteins that can do this, because if not, then you could never change your GTP to GDP. So you would just have signaling all the time, and that could be very, very terrible because, like we've gone over a couple lectures ago, a lot of cancers have something like this, where you have RAS that just loses its GT pace activity. So it is always on, and it's always telling the cell to proliferate, make more of me, and then that's just one step leading to cancer. This slide shows a good example of where you can plot things, whether they're downstream or upstream. So if you have mutant protein X and mutant protein Y, but you don't know exactly where they are on the cell, on the signaling pathway, you know that RAS is at some point involved in this, and you want to know where they are in relation to RAS. So for both of these, just having mutant protein X and Y, you have no signaling. But if you introduce overactive RAS to this cell, signaling is restored. So with mutant protein X, you get overactive RAS, signaling restored. mutant protein Y, you do overacting RAS, and you still have no signaling for some reason. Well, now you can basically say that X works upstream of RAS, because even if X is bad, if you have good RAS, you can get the rest of that pathway going, which is great. But if you have mutant protein Y, which is then downstream of RAS, it doesn't matter how much RAS you have, it can't do anything with it, so then you won't have any signaling. Yeah. Okay, we'll go back. So just remember to look at the filaments, microtubules, and actin filaments. Actin filaments help with protrusions. Microtubules transport things like organelles and proteins throughout the cell. Intermediate filaments make a meshwork, which kind of helps with the flexibility of a cell and how much stress it can handle. And there we go. Microtubules have dynamic instability, which means they can be built as long as they have sections with GTP bound, and when you don't, the microtubule will break down very rapidly. And as you can see here, this is dynin and kinesin transporting things along microtubules. Kinesin goes to the plus, dynin goes to the minus end. And this microtubules in things like cilia and flagella, which help with bending, because as you can see, these microtubules aren't on their own, so they're kind of bound together. So that way, if you have dyning that's moving along them, one will shift according to the other, but since they can't continue to do that, it'll cause a bending. And now exam 4, which we'll go over really quickly, but you all have gone over this a lot, so it should be really quick to remember it. With confocal microscopy, the highlight of this, I'd say, is probably remembering that you receive a high energy photon, the cell will receive a high energy photon, and then the output will be of a lower energy, because you're never going to be able to emit all of what you take in. And this is just receiving light here, so this would be high energy light coming in, hitting your sample, and it would emit a lower energy photon. And this is an example of like a microscope where you have a stage that can move up and down, which you use to focus, because depending on where you're using your, what point you're trying to focus on, it'll have a different clarity at different points. And so CRISPR-Cas9 will use guide RNA to find a sequence. You see you have your target and PAM site, which makes a double-stranded break. And if you don't have any template strand with it to help with that recovery, then you will sometimes get full recovery, where it will look just like it normally did, or insertion of random nucleotides, some deletions, which will cause a frame shift, which just basically means that your DNA won't be read anything like it was read before. Or you can put in a template strand, which is just basically a partner to where your DNA can say, okay, you look exactly like me, I'm going to copy you, which helps for inserting different genes. Your cyclins, oftentimes, as you see, like going through here, it's a cascade of activating cyclins. So subcyclins won't be turned on until others are. So for like cyclin E to be turned on, cyclin D needs to be there, and for cyclin A, cyclin E. And this is a good example of P53, where it recognizes DNA damage. So P53 is super important, because if there's something wrong with P53, you won't ever recognize cell damage. But if you have working P53, then it can see cell damage, and it can basically hold up these cyclins. So to halt the cell and say, hey, it's time to take a break, don't proliferate, because we need to fix this before we go on. If you can't fix any of your damage, then the cell will go through apoptosis, and this is an example of the intrinsic pathway, where you have cytochrome C from the cell's own mitochondria going out and matching with these adapter proteins, which then call procaspase-9 to it, which creates this blade, which we refer to as the wheel of death, which chops a whole bunch of things up in the cell so it can't function anymore, and then it kind of bubbles up and gets eaten by other cells. Or you can have something like an immune cell that comes to the cell and hooks on, and it recognizes that things aren't right. And so there's this little fast death receptor that is separate from the intrinsic pathway, which is why this is the extrinsic pathway, that says, all right, it's time for you to die, buddy. And it'll signal through, which these are also caspases, but they're different caspases, which still lead to apoptosis. Cancer is bad. benign tumor is basically just rogue cells that haven't had the ability to migrate into the bloodstream and get to other parts of the cell. Once they do gain that ability, they become malignant. You can go to this slide. So just some of the hallmarks of cancer is that they keep growing no matter what, and they don't respond to other signals to not grow. So insensitive to anti-growth signals. They evade apoptosis, so they never want to die. They replicate a lot and really quickly. They call blood cells to them, and then eventually they invade other tissues. And are we out of? Okay. Oh yeah. Oh.
So we're out of time, but these slides are posted in your Unit 3 and Unit 4. The full PDFs are there. And of course, are the 1 and 2 slides up?
Yeah.
1 and 2 are in Dr. Lynn's folder. And so check those out, and of course, come to our office hours if you want to hear more. Okay. Good luck on your exam on Thursday.
If you have any questions, email me or come to office hours tomorrow because I have them from 9 to 11 like I do every week.
And I have office hours right after class. Come see me.